molecular genetic networks that regulate plants' resistance under abiotic stress, such as drought and salt. The knowledge generated from the basic research program is being translated to devise genetic tools that would complement the conventional breeding efforts towards developing crop varieties, improved stress tolerance. One of the major mandate crop wherein he has contributed immensely is sugarcane, wherein using not only marker-assisted breeding but also through genome tools, he's trying to manipulate various key traits of agronomic importance. He has also contributed significantly in the crops sweet wheat, sweet potato, and cotton. And he has also been awarded through various grants and awards for his immense contribution, which I believe Dr. Satyendra will elaborate. So my association with Dr. Niranjan has been during my brief training at International Rice Research Institute in the year 2003. And I found him to be a very friendly person, very joyful, and also very encouraging person. And I personally enjoyed my association. I always enjoy my association with you, Dr. Niranjan. And I now request Dr. Satyendra to give the introduction of the speaker. But before that, Dr. Niranjan will be speaking on a topic which is relevant with regard to rapidly changing climate or climate change. He'll be speaking on integrated approaches towards development of rice varieties for alternative irrigation systems. So I now request Dr. Satyendra to give a detailed introduction of Dr. Niranjan.
Thank you, sir. Good morning, everybody. I feel privileged to introduce Dr. Niranjan Basak among you. I think already our director, sir, has given some part of the introduction, but then it's formally I will introduce Dr. Basak. In fact, I am one of the beneficiary of emerging from his lab, and I was trained while my stay at Louisiana State University. Dr. Niranjan Basak, he was born in a small village of Odisha. He finished his undergraduate study in agriculture with specialization in plant breeding genetics in 1990 and MSc in plant breeding and genetics with specialization in tissue culture in 1993. Both from the Odisha University of Agriculture and Technology, that we call OUAT. While he was working in a project with Dr. G.J. and Rao at Central Rice Research Institute, now it is National Rice Research Institute, towards his doctoral program, he received the prestigious Rockefeller Foundation PhD Dissertation Award to conduct research work with Dr. Karabi Datta and Dr. Swapan Datta at International Rice Research Institute in Philippines, where he developed and characterized transgenic rice with seed blight and yellow stem borer resistance using both biolistic as well as agrobacterium mediated transformation. And for the information of the house, I will say till that time there was no published success story of agrobacterium mediated transgenic rice. After finishing his PhD in 2000, he continued at IRRI as postdoc, working in multiple projects, including as a project scientist in USAID funded project that was on golden rice. In 2004, Dr. Basak moved to Louisiana State University Agriculture Center for another postdoc. And there he worked in the coastal plants genetics program until 2007. From 2008 onwards, he has been working as faculty at LSU Egg Center, researching on various model plants and agricultural crops towards understanding and improving stress tolerance using various molecular biology, functional genomics, and molecular breeding tools. He has graduated 7 students as their major advisor and co-supervised 8 graduate students. He has also mentored 12 postdoctoral researchers, 13 Borlaug fellows, and I am one of the Borlaug fellow trained in his lab, 2 Fulbright scholars, 16 visiting researchers, 5 research associates, 8 undergraduate students, and 3 high school summer interns. Dr. Basak has more than 70 peer-reviewed publications in various high-impact journals and secured more than $5 million for his research program at different federal and commodity crop boards. Beside this formal introduction, since I have worked in his lab, I would like to say he's a wonderful host, excellent researchers, and I acquired a lot of his skill and knowledge while I was there. So thank you, Dr. Baisak. And I request now Dr. Sunita to moderate the session.
Warm good morning to all of you. Thank you very much, Dr. Satinder, for the nice introduction for our wonderful guest speaker of today's webinar, Dr. Niranjan Baisak, Associate Professor from Lausanne State University Agriculture Center. This is the part of, as Kalyani has already said in her welcome address, that this is one of the seminars that we are being organized in association with the Society for Advancement of Rice Research, together, Indian Institute of Rice Research and our society. So far, we have organized around 20 seminars, both offline, online, and as well as in hybrid mode. And today is the 18th webinar that we are organizing in our institute through online mode. The webinar is on integrated approaches towards development of rice varieties for alternate irrigation system. Sir, I invite you, Dr. Niranjan Baisak, sir, to take over and then continue your speech. Thank you.
Okay. First of all, I would like to thank Dr. Sundaram, Dr. Satyendra, for that nice introduction, and then also thinking about me to give this talk. I'm very privileged to speak to the rice audience, the real rice audience, and then so I'm pretty sure that I will be lacking in several aspects. So from the very beginning, I definitely beg pardon that if I'm misrepresenting any of the facts here. But anyway, I would go ahead, and again, I'm sorry for this delay and keeping you all waiting. I have a lot of nice memories with many of you. I think Dr. Padma Kumari is here. It goes back a long way, and with no further ado, let me start. Let me share my screen here. So, can I share my screen now?
Yes, sir.
Right. You can see my screen in the presentation mode, right?
No, sir. We are yet to see it, sir. Yeah.
Okay.
Yeah, we are able to see now.
Okay, good. Thank you. Right. So, okay. So they said that my topic would be integrated approaches towards development of rice varieties for alternative irrigation systems. I know during my introduction, I have been known mostly as a plant molecular biologist, working with transgenic rice or gene cloning and functional expression. So I'll just give a little bit of a different kind of talk that we all know. So what's going on? Okay. For some of you who are not really aware of Louisiana, let me tell you that we are actually in the further south of the United States of America. And Louisiana is a very unique place, apart from what you hear about hurricanes in Katrina, the famous ones. But Louisiana is very uniquely placed. As you can see from this map, this is actually the crop map of Louisiana, or agriculture map of Louisiana. This is one state, unique state, where you can grow almost everything. So starting from rice, sugarcane, all the horticultural crops, grain crops, corn, soybean, and even the other row crops like cotton and corn. So as you can see here, we have like 5 different agricultural, regions or egg center regions that are responsible for all of these crops growing. And these are only the field crops I have listed. Apart from that, we have horticultural crops and then animal husbandry and everything. Just to give a little bit of introduction about Louisiana. So now coming to rice, I think these facts are very much known to all of you. We all know that around 1,500 liters of water is required to produce 1 kg of paddy rice, right? So on an average, almost 43 or 40% of the global irrigation water goes only towards rice. And of that, just imagine that 30%, up to 30% of the global fresh water is consumed only by rice. So in Louisiana particularly, rice is very much grown under flood irrigation because we have plenty of water in Louisiana, and I will talk about it a little bit later. But it is very expensive to keep this rice field flooded, right? Both from the cost of pumping is very expensive, and also the fuel that is needed to power them. And then now, needless to say, that the cost of fuel is going like crazy now, right? So we were somewhere around 1.98 in gallon to almost $4 and $4.50 in gallon, so more than double. So, and in addition to that, we have this alluvial aquifer, which is actually going down in the Mississippi River Valley, and that's what actually is causing the shortage of fresh quality water. Okay, so even if we have actually enough water, but then the quality of the water is going down. And another example of this problem is in Louisiana. As I said, we encounter a lot of hurricanes, at least one in a year, maybe on an average. And every time there is a hurricane, we would have the seawater inundating to the rice area, and that makes actually the land unarable. So even if we have plenty of water, then either the salt becomes alkaline or saline because of these hurricanes, right? So you all are actually now aware of the recent drought. I mean, recent means in 2014, California, and 2016, Texas, and then mid-Southwest, we had a severe drought problem. So drought, even if in US, maybe people are, you all think that US is actually very, input intensive, and water is not a problem, irrigation is not a problem, but it is actually a problem. In one way or other, it is a problem. So water has not been anymore a renewable natural resource. It's very, very precious. So, and why this is happening? So what's problem actually the water now? That because of this climate change, right? We cannot deny the fact that there is climate change. We have been having this climate change from time immemorial. Maybe we are just aggravating it. So anyway, there are, as you see here in the map, there are actually two schools of thought though. The carbon dioxide is actually the main culprit for this climate change, right? So the two schools of thought is that if we have more carbon dioxide, then the plants will utilize carbon dioxide to its advantage, and they will produce more. However, if you see this map, that really doesn't hold good. It's only in the northern part, northern hemisphere, where actually you don't have enough of the sunlight, and there is less of the temperature is down. So that's where probably the carbon dioxide is playing a little bit of positive role. But in most cases, it's actually responsible for the decline in the agricultural productivity. Okay. So what is this global climate change is bringing to us, especially for rice? That because of this, there is actually altered timing and magnitude of the water or the precipitation that is either causing drought or flood. And obviously there is global night temperature increase. We know the impact of the high night temperature, and that also can have dramatic impact on the disease and pest incidence in rice. In addition to that, we have this UVB radiation, and then the enriched carbon dioxide can also alter actually the competition between the weeds, and then the interaction between the rice and then the major weeds. And also it can affect to the beneficial organisms, for example, like the nitrogen fixing bacteria or the nitrogen fixation in the rice fields, right? So all in all, this actually translates to that we need to produce more rice to feed the ever growing rice consuming population in the world. So here is actually the rest. This is actually already a little bit older, 3 years older, but you can see here that drought is very, 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 very epidemic, right? I mean, it's completely like everywhere. So whether it is low to medium or to acute stress of water, it's all over the world. It's not just only endemic to only one pocket in the world. So water is a problem all over. And now what drought causes to rice? We all know the impact of drought to rice. Seasonal drought is a major threat to rice production, and especially, as you know that this is a very old thing, I still like to say this one. Aki Kamoshita from IRRI, when he was doing his PhD, so during this time that he published that during, if the reproductive stage there is a drought, what we call as terminal drought, then almost 50% crop failure, and even later on Arvind Kumar's publication said it can also lead to the total crop failure if the drought coincides with the green filling stage, right? So this just to give a little bit of background, in Louisiana, as I said we don't have a deficit of water, but as you can see in some months, particularly I just put only few months in the years where actually the rice is grown. Rice is grown in the period of April. The planting starts mid-March to first week of April and goes through August. So this is actually an interactive map where you can see that even if we do live with good water system over here, but there are actually some belts in southwest regions will have in April, there will be some drought. Even at moderate to high level of drought, we can still experience in our rice growing regions. Okay, so what are the ways? So we all are aware of this mechanism. So in addition to the continuous flooding, so to save water, there were 3 different types of alternate water management systems were projected or being adopted. So alternate wetting and drying, where you actually would be keeping it flooded and then let it reside, let the water reside. And then whenever the water is again down completely, the field is dry, then you again flood it. So it is kept flooded, but it is intermittently flooded. And the other one is actually row rice. In Louisiana, this is actually something that is being practiced over here. So this is grown, row rice means that furrow irrigated rice. So you irrigate the rice in the furrows, water in the furrows. And then grow the crop in the rows, just like you grow corn, soybean, or cotton, or any other grain crops, right? So this is actually becoming very popular in Louisiana, especially in the northern part of Louisiana. As you can see that for watering, so these are like the poly pipes running around the field, and whenever there is actually need, then you just water in the furrows. And then the other one is actually called semi-aerobic, or what we call in our Indian context, the rain-fed. It's mostly rain-fed, but we do actually give the flush irrigation, like we just flush the field, never keep the water standing or in the furrows. It's only just given whenever we see acute drought symptoms in the plants. So that's what we call it semi-aerobic or semi-rain-fed condition. So just to give here, this is actually the slide that I have borrowed from my agronomic collaborator. You can see here how much water actually we are saving. This is delayed flooding, alternate wetting and drying, and then the semi-aerobic condition where we are keeping the field dry mostly until the crop shows severe drought symptom, and that is when actually we do flush irrigate the soil just to saturate it, to keep it moist. So as we can see here, like around 15% grain yield reduction because of this reduction in water use, but we are saving the water by 60%, up to 60%. So if you look at the cost benefit ratio, if you can save the water by even by 50% at the cost of 10% to 15% crop yield reduction, I think the farmers would still go for these management systems because of the costs involved with the irrigation and pumping and then also the fuel to irrigate the field. Okay, so and these are some of the varieties that have been tested for this condition. So what do we need? I guess mostly this rice has been historically grown as a semi-aquatic plant, right? So most of the inbreds that we have actually developed, they have been mostly for irrigated condition. So that's the reason they are not suitable for these alternate water management systems. And what we have seen in Louisiana condition, that the hybrids seem to suit better as raw rice or in this rain-fed condition, just not because that they do have the drought tolerance, just because of their vigor. Okay, so they can actually compensate, because when we have inbreds, we don't have so much of tillering, but the hybrids, because of their heavy tillering and nitrogen responsiveness, they do actually compensate for the number of tillers, by the number of tillers, and then so on. So I guess the recent breeding efforts, because of these recent breeding efforts for the last few years, have been directed toward development of varieties that would have high water use efficiency. That means the varieties must have the ability to tolerate drought, at least periodic drought or prolonged period of dry spells, right? So that they could be suitable to these alternate irrigation systems. Again, like whenever we are dealing with this water, that means there is a stress because of this alternate water management system or this raw rice semi-aerobic. So there is a stress. So, but of course, all forms of life have evolved strategies to adapt to stress, right? So the animal system or the human, we have our system to adapt. We can escape or we can avoid and we can tolerate. So the plants also have developed similar things, although they cannot just escape the way here, the stress. But then they do have all these three mechanisms, even if they are actually not mobile, right? It's a silent organisms. So because of, we know that there is definitely a vast majority of these variations exist in our primary gene pool of rice, whether you have, wherever you have your germplasm. I know that you all have done these works. So you do know there is, of course, a lot of variations that is existing in the primary gene pool, right? So this is actually the rice diversity panel that was from Susan McCoos that generated this. Collected all 420 rice varieties from all over the world, maintained at Arkansas. So we collected those and then screened them in our field drought condition, and to select out some of the potential donors, right? So, and also we did that one at the vegetative stage, stress and then see. So this is actually Bandhana and N22. These are the two Indian varieties you all are aware of. We have been using these two varieties for introgressing their drought tolerance abilities to our high yielding varieties like cocoa tree. Okay, and so we have been using a lot of different populations for screening these materials. So what we are doing is, there has been a lot of progress in, I have just only listed the ones that IRRI, but I'm aware of varieties that are being released by the Indian Institute of Rice Research, or also by my friend Dr. Pradhan at National Rice Research Institute for the aerobic conditions or semi-aerobic conditions. So what we need to develop, but again, the varieties that are, you all know that in the US or in Louisiana, we grow actually rice which are mostly tropical Japonica, right? So the indica varieties that are drought tolerant that are being developed in India, they do not just automatically suit to our system here. So we need to introgress those things to our locally adapted varieties. And then the idea is actually to combine high yielding traits of our varieties with the ability to tolerate periodic dry spells from those exotic germplasm. When I say exotic germplasm, means from India or elsewhere. So these are actually the populations that we have developed, and then we are screening for identifying the potential lines. Sorry. So on the cocoa tree bandhana area recombinant inbred lines, then cocoa tree by N22 recombinant lines, the rice diversity panel that was actually number 1 before 420, and then we have now the 560 rice diversity panel 2, which contains more of the Japonicas, and also the US rice breeding pedigree lines. So we are looking at the pedigrees of the US rice breeding, the modern rice varieties of the US, so that we can see if You all know from the publications from Arvind Kumar's group that with the development of more of the semi-dwarfing rice varieties, because the linkage between the SD1 and then the drought tolerance has been broken, right? So that's the reason most modern varieties are drought susceptible or drought sensitive. So we were trying to look at actually the pedigrees, that if there were actually drought tolerance genes in the US pedigree lines, so that can be also brought up in addition, instead of just only bringing those genes from indica, because you all know the potential problems that are associated with indica japonica process. In addition to sterility, then we also deal with some other linkage drugs, for example, some disease and then other stuff. So initially, we would screen them in the greenhouse condition, and these are very acute vegetative to reproductive transition stage. I particularly prefer at greenhouse conditions because it is very acute. So I would not give them at the terminal stage. I would give them just at the pre-booting stage. So when there is a transition from the vegetative to reproductive stage, and then we select out those, and then we grow like this. As I said you that we do not, in Louisiana we have more of the water compared to Texas, and then we delay the planting to coincide the summer with this with our crops, so that we can screen them under the field condition in semi-aerobic or rain-fed system. As I said you that they will never be flooded. Only when we will see acute drought symptoms, then we will be just to flash flood this field. Okay. So this is how we do, and then we would be screening out some of these lines and from the recombinant inbred lines, and that we have selected out, and then we would grow them in the row rice system at Crawley in collaboration with my agronomist friend Dr. Manok Kongcham. So, and then we also calculate the amount of water that is being put into these fields. And this is actually, I think I'm just trying to show you. So these are actually some of the gas traps. So we try to measure the carbon dioxide, the methane, and then the nitrous oxide from the rice fields. Both, so we do have the flooded fields over here, and also from here, to see actually how we can reduce also the methane gas emission from the rice field by using this alternate water management system, scheme or system. And then whatever we have been already identified as potential lines, we go again for purification and seed multiplication. So these are some of the potential lines, and just to know that we try to look at the lines that are long grain, because US rice is mostly focused on long grain, particularly in Louisiana. So we try to select out those which are especially long grain rice varieties. And then some of these things are very promising. So we have been actually working on these things to see if we can potentially make some of these lines, or at least a couple of these lines to the release date. So these are some of the cultural issues although to consider, right? So because rice has been grown historically as a semi-aquatic plant, so the weeds are typically smoothed by the standing water. So when we are not having standing water, there is a problem of weed. So herbicides with overlapping residual coverage is actually the best strategy. That's what actually we try to follow it in the Louisiana conditions. And we all know how devastating blast can be, and especially when there is drought stress. Blast is pretty much kind of associated with drought. So wherever the field is dry, blast takes over. So obviously there is a little bit of increase in the fungicide, and also whenever we screen the varieties or the lines for drought tolerance or for their suitability to alternate water management system, we also look for the score for the blast resistance. And the nutrient management again. So we all know, because under dry conditions, so there is actually chance of leaching whenever you actually flash flood. So there is one dose of or one more application of nitrogen fertilizer we make it during. So basal top dressing, and then there is one more third application with 40 to 50 kg of urea per acre of rice field. Okay, so those are the things that I told you, that because we do have populations, so we actually, because I'm a molecular biologist by training, so my objective is actually to identify what actually makes the plant or some of the varieties drought tolerant, or how we can actually understand the mechanisms of drought tolerance from these varieties like Bandhana or N22. There have been multiple studies done, but not actually in the background of the US rice germplasm. Because as we know that most of these genes, they would actually act differently in different genetic background, right? So this is a study that to understand the genes that are being differentially regulated between cocoa tree and bandhana, and also cocoa tree and N22. I've just given example of cocoa tree and bandhana here, and Dr. Satyendra was involved in part for this research as well while he was here in 3 months in my lab. And this manuscript is actually about to be submitted for publication pretty soon. So there are some genes that we can see that these are the unique genes, like in 273 genes being upregulated differentially, and then 364 genes being downregulated over here in bandhana. And then of course there are common genes, and these genes are very conservative numbers because whenever we select out these genes, these are very, log 2 fold 2. So and what is the objective is actually to, when we looked at these genes, then we also look at the structural variants in those genes, so that we can look at what are the SNPs or the simple sequence repeats that are existing in these genes. This of course map is showing different structural variations with among these genes that are unique to bandhana or unique to cocoa tree. Okay. So, and also because we have so many genes, potentially we cannot actually select all of these genes for functional validation, like I'll talk a little bit later. So we also look into the gene co-expression network analysis to look at what if these genes are actually, as you see that the genes are actually separated here by the modules are whether it is the photosynthesis or transcription factor or calcium signaling genes. We just want to know, and then the idea of doing the gene co-expression network, you all are aware, to identify some of the major hubs that are having maximum connectivity with other genes that we can potentially target during our transgenic or gene editing approaches. So at this moment, we are only focusing right now with this the structural variants, because we will be using the structural variants for mapping the QTLs. So we used those mapping populations, and this is actually just the again, I think I need to mention here that our trait is mostly and only grain yield traits, because if I follow up or if you remember the publications from Dr. Kumar, that yield is the most inherited trait under drought stress. All other traits, of course, there are secondary traits like the photosynthesis, chlorophyll content, and all those things will be factored into the yield. But then grain yield per se, or the yield traits like the direct component traits like panicle or the grain number, the grain weight, these are the ones that are mostly inherited under drought stress. So we've been focusing only on those traits. Okay, so these are some of the QTLs that we have identified in these two populations. I will not get into the details. In addition to the QTL in chromosome 1, which is actually known from IRRI, the QTL 1.1, that has been very well known at 1.1 from IRRI studies, and then a lot of studies from India as well. So in addition to that, we have identified the QTL in chromosome 3. This was also reported by Mallikarjun Swami, but then as a meta-QTL. So we also identified this chromosome 3, 5, 8, and 9, and the 3, 5, and 8. There were some of these QTLs, chromosome 1, 5, and 8. These are actually consistently expressed in both the populations under stress. So I just want to say here one thing, like when this green color, these QTLs, they are actually the green color grain yield under control. Okay, so the green color QTLs are under control. So that means And the grain yield under drought stress, those QTLs are definitely independent of the QTLs that are under, for grain yield under control condition, except for this particular QTL over here in chromosome 1. There is a little bit of overlap if the confidence interval is taken into account. And and also the panicle number under control condition is overlapping also with the grain yield in this particular condition, in this particular chromosome 1. So these are the three chromosomes that are actually consistent between the two populations. In addition to that, I told you that the chromosome 3 is actually the one chromosome 3 QTL is one of the QTLs that overlaps with the meta QTL from Dr. Swami, and then I had actually some extended talk with him. So this is, again, I'm not going into the details. These are the allelic variation at those particular locus for the grain yield. So again, here I was telling you. So this is actually the meta QTL over here. The two of these QTLs that is encompassing the two meta QTLs that were identified by Swami. Okay, and I'm focusing on this chromosome 3 because I will be talking a little bit later. So the genes that are actually in this region in chromosome 3, this is actually again a protein-protein interaction, predicted protein-protein interaction. And in this particular interaction, there is one hub that is actually a alternate actin depolymerizing factor. And again, I will tell about this one because we just have published that the actin depolymerizing factor from spartina, which is again a smooth cordgrass, and that actually contributes significantly to drought tolerance in transgenic rice. So this is one of the QTLs or this particular gene we have actually patented because we know that this gene is very much a player in this particular QTL. It's not actually physically mapped, or I have not cloned this QTL, but we know this gene is there, and from our study we have seen this gene is significantly contributing to drought tolerance. So in addition to just using the primary gene pool, we have been also using in my lab in parallel some of the extremophils. You are all aware of these different extremophils like Thilungiella. These are mostly halophytes, but not necessarily they only tolerate salt, especially for crops like Spartina alterniflora. Plants like Spartina alterniflora, which is cordgrass. It grows in the Louisiana coast. It's a halophyte. In addition to that, whenever the coastal land is dry, this plant also still stands. And then we also have noticed that Potacia also can survive low level of drought stress. Okay, so we have been using these two particular plants in my lab, and our strategy is actually to identify the genes through a full-length overexpressor strategy. So we made the libraries, all the libraries of this particular Potacia or Spartina, and then this is kind of a library screening, right? So we screen the whole library in agrobacterium and then transform to Arabidopsis or rice. And then we screen those rice or the Arabidopsis for drought or salt tolerance. I will not get into the salt. My focus will be mostly on drought. So, and then these are actually the rice transgenics that are overexpressing the genes from Potacia quercata. And then there are some, this is just one representative picture I'm showing. So these are some of the rice lines that show very nice drought tolerance under the greenhouse condition. And you can see here that after 14 days of stress, this is the control plant or the wild type, they're completely fried. And there are some of these plants are still standing. And again, like these are a lot of plants that were in the initially 3 days, 5 days, they will be surviving, but by 2 weeks, most of them are gone and these are only surviving. So these were some of these plants, some of these genes. So again, when we actually identified these genes, one of these genes was again matching to the actin depolymerizing factor. So what is this actin depolymerizing factor? I just want to give a little bit because this gene is very close to my heart. This actin depolymerizing factor are these proteins. They maintain the actin dynamics by maintaining the balance between the F actin or filamentous actin and then the G or ground actin, right? So what happens under stress? The cell actually experiences and then the actin cytoskeleton gets disturbed. And that is because the filamentous actin does not get depolymerized very well. And instead of depolymerization, it gets fragmented because of the stress. So the G-actin pool is not enough to again repolymerize to F-actin. So the balance is disturbed. And that's the reason the cell collapses. But in this kind, particularly like a grass like cordgrass or Cortesia, this particular gene is very highly active. And we have shown from, I would not get into the details, we have published this work through my postdoc researcher, Sonali's work, that we have seen that this particular actin depolymerizing factor is less phosphorylated compared to the endogenous actin depolymerizing factor in rice. So because of this less phosphorylation at the serine 6th position, the serine 6 is actually replaced by threonine in this particular gene. So that's the reason the phosphorylation is low and the activity is high. Because of the high activity, then there is higher depolymerization. And that's the reason that the gene is very highly active. So, and again, as you see here, this gene, whenever it is overexpressed, this is 7 days after stress, this is the wild type, and then 14 days after stress. And whenever we gave the water back and then with the recovery, then the wild type could not recover. And this is the same thing again with the grain filling under greenhouse conditions. All these things are under greenhouse conditions. We do have now the EPA permit to get into the field for field testing. In fact, we are actually having the poly houses because we are building actually the polythene on the sides to somehow cover to make this pollens restriction, because we have a lot of rice in Louisiana, so we cannot take those chances. And in addition to the above ground yield, we also see significant difference with the underground root system in these transgenic lines, like these three are the transgenic, this is the wild type. In addition to that, this gene also, probably because it's a halophyte gene, so it is also contributing significantly to salt tolerance. This is a float leaf assay, and then also in the hydroponics. And this, as I said, that this gene we have also have our intellectual property on that. So I will not get into the details. These are very conventional stuff that whenever there is a transgenic work, then you would show that these transgenics are accumulating less of these superoxide or the hydrogen peroxide by this biochemical assays. And one thing I would like to stress here that the common notion is whenever a plant is under stress, the stomata closes, which is very understandable because the plant would like to, or the cells would like to, tissue would like to conserve water. Unfortunately, the longer the time the stomata closes, the plant actually experiences, it has some negative effect on the photosynthesis because there will be less carbon dioxide exchange, right? So this particular transgenics, like this is actually one of the rice homologue, which we found out that it is also almost equivalent to spartina gene or the Patricia gene, where actually under stress condition also the stomata is wide open. Okay, so that actually kind of makes that even if the stomata is not closed, but still there is a tissue tolerance. And that's because of the higher activity of this depolymerizing factor. And we also looked at that also in the chloroplast structure, that the chlorophyll or the granular structures are very well maintained in the overexpressing lines. And then of course we have the RNA lines where we see the granular structure is completely messed up over here, not like the layer structure that you can see typically. So this is just to show these are actually actin mesh. Typically, you will see the actin like this as a actin network or mesh we call, and then under stress, you would see like the actin is actually defragmented. This is not like depolymerized, right? So that's the reason you would see like the chunks of these actins, they are not really able to repolymerize again. But here under this or in the overexpressed lines, you can see that they are still actually very much in the mesh structure, right? So there is no defragmentation marks, and then they are still like filament structure. So, and then in the RNAi lines, you can see that they're actually defragmented again. Okay. I would not get into the details. So I told you that there are two things in this particular gene that we have targeted for the gene editing, because there is one serine threonine change over there. So we have tried base editing to change the serine to threonine, and as well as there is a difference in the F loop structure in this particular actin depolymerizing factor. So the F loop is the one which is responsible for binding the actin, binding to the actin. So the more the actin binding property of the gene, the better is its activity for depolymerization. So we have targeted these two regions for our gene editing studies. So we have some preliminary data on these edited plants, and this is something that we are preparing for publication. It's not yet published. So yeah, this is again the same thing. I would really probably, I think I'm probably kind of getting close to my finishing line. So my objective is actually that, or my take home, which is not really something new, but it's a holistic or integrated approach, not only just focusing only on one thing. We probably should be looking into multi-pronged approaches to understand the intricacies of this abiotic stress response, or particularly drought stress response in rice, or any other crop for that matter, and then use those knowledge to translate for improving the stress tolerance in plants. And then of course, with that, I would like to thank the people who have immensely contributed to this work. This is my former postdoc who has done some of this earlier work. Sonali did a lot of work with this actin depolymerizing factor for biochemical characterization, and Jonalyn, especially in the field and greenhouse works. She has since moved on to Texas. And Nia, who used to work, and then I do have a couple of postdocs now that I did not put here. They just came in, and then continuing with this ADF gene editing work. And also some of these collaborators, Andy Pereira from Arkansas, Adam, and then Manoj and Dustin from LSU Ag Center here in the rice station. And Dr. Tabian, he has retired from Texas, and then I'm working with Omar Samantha now there, and also the funding agencies for making all this work possible. Thank you very much for your patience. And I would be happy to take any questions that you have.
Thank you very much, Dr. Niranjan, for giving a very good glimpse of various approaches, starting with basic genetic approach, wherein experiments are done at field level, and then moving towards using some of the molecular approaches, using molecular markers, and then later using some of the cutting-edge genomic tools and transcriptomic tools to understand the intricacies associated with drought tolerance, and then finally culminating with transgenics as a tool for abiotic stress tolerance. The talk was wonderful, and then we all really enjoyed it. Thank you very much, Dr. Niranjan. Before we start a formal question and answer round, I have one pressing question in my mind. Of late, everybody is sort of going gaga over genome editing. And it's my perception, I may be wrong, that while genome editing is very important to improve some of the native traits of rice, or some even novel traits can also be brought in. But I would say that it should not be genome editing vis-a-vis conventional transgenesis approach. Both are mutually complementary. What are your views in this particular regard, Dr. Niranjan? You have been working in both the fields for a long time.
I completely agree with that. And that is probably one of the reasons why the private industries have not shut down their transgenic program, because there are certain limitations in addition to all those regulatory and other aspects. There are also certain technical challenges with gene editing, especially, for example, like a crop in vegetatively propagated plants. So where it would be very difficult even to separate out if you are using particularly the SDN or SDN2 type. In addition to, so that's the reason you probably would be going for more RNP mediated or some other type of things, or DNA free gene editing approaches. But in addition to that, it is just not possible for all and every trait or every gene. We have to be looking. I mean, we have looked. I'm sure Dr. Mangrothe is working. I mean, and a lot of people are working. There are certain genes that we just cannot find the right place to target. Okay, so that is where actually we will fail, or we will not be having any kind of way to go about and then edit it. Okay, so and that's the reason some of the things like transgenics would come in play. The same thing like when we grew up or when we studied, we knew that there is no definitive strong source of resistance to yellow stem borer. Before, of course, in your institute, Dr. Ram came up with very good sources of resistance. But that time, transgenic was the only approach, right? I mean, only way
Even now
to.
also, I would like to correct, sir. Even now also, there have been very good sources, mutants and all. As on date, I have analyzed the data. Transgenics is the only approach to manage stem borer. It's looking at half glass empty or half glass full with regard to the other approaches. Transgenics, according to me, is the only approach through which we can very reliably or very significantly manage stem borer.
Yep, I agree with that. And again, like, I stay here in US, and then we probably grow most or the maximum amount of transgenic crops, right? So rice is, if you ask me this question, I know many people ask this question, that why not rice? We wear actually transgenic cotton shirts. We eat transgenic GM soybean oil or GM cotton, tomatoes and everything. So rice, why not rice? Because US rice market is mostly for export. And the market is mostly Japan and Europe. So we cannot mess up our rice market. That's the reason it is not done. Not just that because we don't like transgenic rice here in the US. It is because that we do not want to mess up the export market. And both Europe, we know that European Union and then the Japan, they are actually not against, I mean, they are not actually in favor of GM rice or GM. So that is the only reason that we don't do rice. But I mean, for countries like India or like, I know, I'm so glad that the golden rice is now being commercially cultivated in Bangladesh, and then it will be soon probably in Philippines. So it's not too far. So we should not just forget about transgenic. And gene editing is not going to completely replace the transgenic. It is another approach. So just like another technology. So yeah, we should not be completely reliant on just one technology.
Thank you, Dr. Niranjan. You clarified a lot of things. And now I request the moderator for today's session, Dr. Sunita, to go ahead with the question and answer session.
Yes, sir. Thank you very much, sir. Really a very in-depth presentation, sir. I myself like getting involved in many of these things, and I have a lot many questions. Rather, I would like to discuss sometime when I get. But one question I would like to ask you, sir, Dr. Niranjan, sir. You have a rich experience in aerobic rice and in the drought tolerance. Nowadays, the focus is on aerobic rice. So that aerobic rice ecosystem itself has lot many abiotic stresses, related abiotic stresses. So what are your thoughts on addressing the crosstalk among the different abiotic stresses, sir, even in ecosystem?
Okay. I would think that probably the only other abiotic stress that I would associate with aerobic rice would be heat. I mean, we don't, of course, we grow, and then if there is a sudden flooding, then that's something different. But I think the heat is something that is definitely associated with the aerobic condition, because you have also heat from the soil in addition to the canopy, because when you don't have enough water in the plot, so that means there is actually heat radiation also from the soil surface in addition to the canopy temperature. So yes, that is something that we need to be really thinking about when we are breeding for the varieties for drought tolerance or this aerobic rice system. I'm aware of that, and then I think this is one thing that, and it's so complicated that drought and heat they do not have a lot of overlap, unfortunately. There are, of course, like Nagina 22 is one of the varieties that has been implicated or has been shown that it also tolerates heat, but then we still do not know the, or at least I do not know the basics of that particular variety, because to be very honest, Nagina 22 does not grow very well in Louisiana condition. So only in the greenhouse condition that we can test, and it's very difficult to mimic the stress conditions in the greenhouse situation compared to the field. So I think we need to probably be looking at both, integrating both drought tolerance as well as heat tolerance for a variety to be more successful, especially in the long growing season like in India.
Thank you very much, sir. We have participants across the rice ecosystems all over the India. So I request our participants to have any questions, please come forward and then interact with our distinguished speaker. Please.
And the questions can also be typed in the chat box so that the moderator
Yes.
can raise these questions. There's one from Kalyani.
We have one question from Kalyani. Dr. Kalyani, would you like to ask, or should I put that question?
Okay, ma'am. Thank you, sir. Thank you for the talk. At what stage the plants are being phenotyped under a drought stage, sir, under drought stress, and especially for the roots, and which methodology has been followed? Can you please give a brief on it?
Okay, so I do not do the root phenotyping at the field level.
Okay, okay, sir.
So the root phenotyping is only at the greenhouse condition. And the stress is, as I said, like it is, the stress is given at the transition from the vegetative to reproductive, just the booting stage.
Okay.
So that 15 days period from, I mean, most of our varieties actually will flower around 85-90 days. So between 75 to 90 days, that is actually the time that we will be giving the stress.
Okay, sir. Thank you, sir.
Dr. Niranjan, I have one question. In India, there is a perceptible, particularly after COVID-19 hit very severely during 2020, there is a perceptible shift towards direct seeded rice with complete or partial mechanization. What is the trend there in USA? Of course, the rice area is limited, but I understand that wherever rice is cultivated, it is cultivated in pretty intensive manner.
It is all direct seeded rice for us. We never have any transplanting.
Okay, okay. So it is complete mechanization or partial, sir?
No, it's complete mechanism. So I think that is probably, I think the aerobic system or the semi-aerobic system would be very good, suitable for the you have now. And I think I saw some news. I always get very excited when I see some private industries in India. Also, I keep track of those things, that they have like a drip irrigation system. And I think I read or I watched one of the, or maybe I attended on, no, maybe not live, but I attended I heard one of the speakers from your, that you moderated or you invited, who was speaking about the drip irrigation in the rice field. So I do not know how feasible that one is. It might be good in a small scale, but maybe in Indian condition, because I know most of our farmers are still not doing the farming at the level that the farmers, and I mean, Dr. Satyendra and you all know how the farmers are here, right? So the farmers actually support our research here. So if that can make sense to you, so the farmers generate that much money that they actually put their tax money, and that is going towards to the researcher, to the scientists. So that means they're actually big farmers, right? Like having 200 hectares or 300 hectares of crop, I mean, land, or maybe more for rice. So I don't know. So that's the reason our farmers in India, they're kind of little bit, I will not say any more marginal now, but probably still kind of I think in a smaller scale, probably drip irrigation may be possible, but maybe when you adopt the dry seeded or direct seeded rice, aerobic, semi-aerobic, or the raw rice would be a good strategy if we have some good drought tolerance incorporated into that. And I have seen several publications from CRI and IRR. There are some varieties being developed for those systems. Yeah.
I think there are a couple of hands raised, Dr. Kalyan. Just
I see Dr. Padmavati there, so it's after a long time that I see her. So
yeah, Dr. Niranjan, how are you? I feel very happy to see you on this online. I still remember.
So the last I saw you was when you were doing PhD with Dr. Kolle. So
yeah, that time you were a graduate. I think you are doing some third year BSc or so.
That's right. So I was doing my master's. Yes.
Yeah, everything is fine.
Yeah.
So, Dr. Niranjan, congratulations for your excellent talk. And here is one submission from my side. When one's rice is transplanted in the field, it is exposed to different kinds of abiotic stresses, be it drought or salt or submergence, whatnot. So many stresses are there, especially in, everywhere. It's globally occurring. So as far as India is concerned, we have a very good variety by name DRR 150. It has got both drought tolerance, having QDTY 2.1 and 3.1 QTLs, and also submergence tolerance, having sub 1. So like this, in your place where you are working in Lausanne University, are there any lines available with rice lines or rice varieties with multiple abiotic stress tolerance?
I would say no, because as I said, our rice cultivation is pretty intensive, but also if you really look at the commercial cultivation, it's not actually under drought condition because we do have irrigated. Just to give a little bit of background. So when I first started this drought project, I approached out our rice breeder. He's retired, and then he's a very eminent scientist with a lot of varieties to his name. He's still working for the Rice Federation. So his first answer was, if we don't have water, we don't grow rice, period. So then forget about your drought research, But because California experienced that drought, Texas experienced that drought, so then they started to realize, the farmers started to realize, and now that they know that pumping irrigation water is so expensive, the fuel is so expensive getting, and then drought is getting imminent also. So I work with drought, and we do have a division of labor here. So I have another colleague, Dr. Subudhi, you probably all know, maybe some of you know, Dr. Prasant Subudhi. He works with the salinity tolerance mostly. So, and there are ways that we could combine this, but we never have at least thought of combining these for the Louisiana ecosystem, but it is possible to do. And then I would be very happy that if you have such kind of varieties, maybe it would be very nice to do some kind of studies to see how that actually plays out. I just keep myself again, like I said, because I'm not a breeder, and I don't want to too much. It's difficult that I cannot deviate too much from my focus. If I am actually getting into too many stresses, maybe it will be more thinned out, and then I'm not actually responsible for developing varieties per se. And just because I do have the genetic resources that I have developed, so that's the reason I'm screening. And my main objective is to understand the mechanisms, and if we can actually go we take a longer route to find out some ways to go shortcut. So rather I will stay that way. So I think I do not work with multiple stress resistance, to be very honest. And then I thought like I would be probably contributing one piece to the puzzle instead of solving the whole puzzle, which is not within my reach.
Thank you, Dr. Niranjan.
Thank you.
Thank you, sir. we
Sunita,
have, yeah, ma'am.
can I take this question, Sunita?
Yes.
Dr. Niraj, a very enlightening presentation. Thank you.
Thank you.
I have one question. You were talking about phenotyping in the glasshouse conditions and field conditions. And finally, you narrowed down to certain regions which are common, but the regions are very few between glasshouse and field. And there are many regions which are promising under field conditions. So what is your take home message? Should we take the regions that are common for both the conditions, or should we look into only the field conditions? And does this protocol vary with the stress we impose?
Yes, that's a very good question. I would still vouch for the one in the field condition, because that's where actually you will be seeing the expression. But I just want to tell that even the greenhouse may not 100% translate to field, but the stress in the greenhouse is more controlled.
Yes.
So what you get for drought stress in the greenhouse condition is very, very representative of drought stress. Because under field condition, you do not know how much the subsoil, there is water or moisture in the subsoil condition, unless you are actually putting the moisture meter everywhere in the field. And also depending on how deep you are going, right, for phenotype or to measure the soil. So if we are, depends again, how many gallons of, or how many liters pot that you are growing in the greenhouse, and then how much water actually you are putting. So most of the, you very much aware of the drought, you give like progressive drought or drop down drought, right? So most of this drought in my case is actually just drop down, because they're never water from the top. Dr. Satyendra has seen in my lab, in our greenhouses, that there is no water actually from the top of the pot. It's always capillary. So when there is a capillary and then you take it out, then it's like acute stress that you just put a drop down. There is no progressive drought here. I'm not stopping the watering. I'm actually just taking it out of the water, right? So that way it creates actually, or it mimics to the field condition, because in the field condition, unless there is rain, then there is no more water, except for the subsoil surface. So if I am actually interested in drought stress, I am more actually interested in the greenhouse stress, greenhouse condition. And if that QTL is expressing in the field, then that is very good.
So you have taken only those QTLs which are common for both greenhouse and this, and then you have gone ahead with the analysis. Is it so?
Correct.
Okay. And my next question, half of it you have already explained. Is there any way you tell the farmers that there is drought, like without measuring or putting water meters or anything, that we can indicate like this is the critical? By default, 14 days here and there around the booting stage, you have taken it, I think. So it varies with the type of soil, I presume, the drought. So if the water holding capacity of the soil is less, is there any other means to tell our people that there is stress and you have to irrigate, something like that, apart from the experimentation?
You definitely can tell, because when there is a water shortage, then there will be leaf rolling, right?
Yeah.
The leaf will start to roll, whether it is an avoidance mechanism or whether it is actually a susceptible reaction. The leaves will still roll. So I think that's the strategy that we use actually for aerobic condition. Whenever we see some varieties, of course, we do have susceptible checks. So whenever we will see that the susceptible checks are having acute leaf rolling, or like what we call like temporary wilting point, and then mid wilting point and permanent wilting point.
Yeah.
Before it enters to the PWP, at the mid wilting point, that's the time we will flush the field. So that is what I would say, that this is probably one of the initial symptoms of the drought stress, that the leaves will start to roll. Again, leaf rolling is not an indication of resistance or susceptibility, that you all know, because varieties like Bandhana will roll the leaves very soon. But we know Bandhana is a very drought tolerant plant. But that already tells that there is definitely shortage of water.
And imposition of the stress to what level in the subsoil will create appropriate results, or will be the critical below the ground? How much water level up to what depth will be really needed to create the right experimental conditions, or to indicate to the farmer that beyond this depth, if you put water also, the crop will not recover? Both ways, for the experimental purpose and for the farmer's purpose.
I am sorry, I definitely don't have that answer. I did not do that in-depth study in the field for that at that level, that I would not be able to answer that. But I can say if I am actually putting a soil moisture meter, then I can say that if it is less than 25% field capacity, I can tell that it's going to be the temporary, the wilting point already. So 50%, 50% below field capacity, you already have like the initial drought, initial dry spell. And then if it is 25%, that means you know that there will be wilting point already. Or 25% is already like drought stress. That's already stress. Yeah.
Thank you. Thank you.
Thank you, ma'am. We have few messages in the chat box from Girish. Dr. Girish, would you like to ask, or should I ask this question?
You can ask. Yeah, sir, thank you, sir. Excellent presentation and good information. I just wanted to know what are the bottleneck traits, particularly in the direct seeding conditions, which you are addressing.
Yeah. First of all, the seedling establishment, right? That's actually the germination and the seedling establishment. So I think in here we do not have that anaerobic condition, which I have seen some publications that in IRRI, coming out from IRRI, that you have to have some anaerobic germination, because after you do the seeding, then if there is actually heavy rain or the water is standing in the field, then you need to have definitely some varieties that do have the anaerobic germination ability or the survival in the anaerobic condition. That would be number one. And also the seedling stays what in the greenhouse condition we have seen this, but also you will see that the pythium or some kind of rotting, right? The seedling rotting. So because the transplanting, you don't experience that, but in the seedling, until it is established, the seedling is established, then that is where I think that's the only thing that I would say in the direct seeding rice, from the germination to the first establishment. And you need to definitely be keeping that time. You cannot really play around with water too much. You need to have the soil definitely moist enough.
Sir, regarding seedling vigor, sir, is there any specific gene which you are or any people are working to target editing or utilize in breeding program? Any particular gene which can enhance the seedling vigor?
I have seen actually a few publications that have mapped QTLs for seedling vigor, but I'm not aware. Maybe Dr. Satyendra, have you seen any gene that has been edited for increasing seedling vigor? I have not come across any such.
Okay, sir. Thank
you, Dr. Girish. We have also a question from Dr. Ananta. Dr. Ananta, are you there?
Yeah.
Yeah, you can.
Sir, it's a wonderful presentation, sir. Earlier, actually, we had means a lot of work they did on exclusively root morphology and study to have the drought tolerance. But nowadays, strategy have been little bit changed, not exclusively on root. Of course, root they are considering how much it is contributing for drought tolerance. As such, root itself is enough, or we need to look into other traits also, sir.
Yeah, and lately I'm a little bit interested with root, not for drought tolerance. Let me tell you, because I have always intentionally avoided root because it is just so difficult to phenotype. And anything under the soil, I know they say like there is the other half of the life is under the soil, but that is true. But there are so much challenges involved with that other half of the life of that we cannot just just doing, but it's very important. Why? We all know that the gene that was cloned by a friend of mine in Yusaku Oga lab in Japan, and the DRO gene, deep-rooted gene. So I have mixed feelings about the root because I think there was a publication long before from Jawahar Ali and Zhigang Li, that when there is a salt tolerance or when salt screening, then you need more of the fibrous roots, right? And then when there is a drought stress, then you need actually more of the deep taproot system kind of thing, the deep rooted system, this thing. But that concept has changed now, because if I actually take my example in sugarcane, the more actually you have the fibrous roots, the better is the survival under water stress, because they can just spread out and look for the moisture, and not only just depending only one or two deep roots system. So I have tried to avoid the root before, because I just did not have the capability of phenotyping. But if you do have the ability to phenotype, I think root system architecture improvement is very important for this stress, and not only for that, but also to study because of this the microbiota around the roots that are the rhizosphere, which are also contributing to several abiotic stress tolerance. So I think it's very important to study the roots. My interest with the root is not with that one. This is one topic that I did not touch here. That I'm obviously interested in ratooning ability, improving the ratooning ability in addition to drought. So that's because of my interest in sugarcane, because in India and then places like in India, Brazil, and then Colombia, sugarcane can be grown almost like 5 to 8 crops, right? One time you plant and then you can ratoon for 8 or 6 or 8 ratoons. So we do not have that ability in Louisiana, because after 2 or 3 ratoons, there is so much of yield decline, then they do not ratoon anymore. So then plow out. In rice, we do have actually some very, very nice mutant lines that actually until 3rd ratoon, there is 25% or 30% only yield reduction in the 3rd ratoon. But I'm interested in those ones, and then what it makes to ratoon very well, especially after the 1st or 2nd harvest. So that's the reason I'm actually looking into the root. And I think that is something that would be very interesting in India. I'm pretty sure that you might be already working, or some of you might be working, because you can grow that. Ratooning in rice is not very important in places like Louisiana, because we cannot grow. Rice is just grown only as one crop in it, one season crop. That's it. We don't grow rice actually more than one crop. Very rarely some farmers grow one ratoon. But in a place like in India or in tropical climate, subtropical climate typically, I think if you can still harvest 60 to 70% of your crop in the 3rd ratoon, I think that there is a worth. There is definitely some potential over there. If you just sow it for only once and then harvest for 3 times, I'm pretty sure there is definitely some potential there. I would be happy to hear actually your expert opinion about that. Maybe Dr. Sundaram or some other rice breeders or science experts can comment.
There have been a lot of buzz over ratooning in rice after a series of paper from Chinese group appeared. But there are only a few areas wherein rice cropping system is taken up, that too restricted to some parts of southern India and then eastern India. So it's a question whether we need to invest to do research or acquire those germplasm, and then whether we need to, because ratooning per se for the health of the soil and also for viability of the ecosystem, it may not be very desirable. It's always better to follow rice with some other crop which has a different mechanism and probably add to soil health. So that is one question. even if we consider ratooning, probably hybrids wherein the farmer spends a lot of money for purchasing the seed and they do have inherent vigor could be one of the choices for going for ratooning. But unfortunately, south of Tropic of Cancer, we have only a few areas where hybrid rice or F1 hybrids are actually being grown. So we are just still watching the scenario, and then we'll try to acquire those donor parents from China or elsewhere. But we are cautiously optimistic with regard to ratooning as an approach.
Okay, I think I understand definitely the concern with this monoculture system particularly, but I'm still kind of thinking that even if you can get in 2 crops, because you very well know that the second or third crop is very fast, right? I mean, even if you are spending 5 months in the first crop, and then the second crop would be like 2.5 months, and then the third crop will be just 2 months. So if you are actually able to expand 9 months or 9 to 10 months, then there is still a window of 2 months to grow a kind of a rabi crop or something like that, a legume crop, to kind of reinvigorate the soil health. Probably, this is again me also. I'm still in the very early stage. I have thought that this is something that is more useful for tropical or subtropical climate like India than, of course, here. But we do have some.
In states of Karnataka and then mainly in Tamil Nadu, wherein they take 2.5 or 3 seasons a year, it could be a very suitable system, because there are certain areas other than rice you cannot grow anything. But such areas are becoming a minority in the country, in the sense that more and more areas are facing water stress. And the general policy of the government is to replace rice as a second season crop and then grow for other soil regenerating crops like pulses or oil seeds, which can bring in more income. And added to that, we also have a typical problem in the states of Telangana and Andhra Pradesh, in the sense that the Rabi season crop, it coincides with earlier we used to experience heat stress much after April, but as of now, for the past 4 or 5 years, peak heat period starts by the last week of March or first week of April. When the crop matures around that time, then there is a very high possibility of reducing head rice recovery, lot of breakage. So farmers are resorting to parboiling, which is not decided by public distribution system. So as a policy, government, all the state governments down here in South are trying to discourage rice as a second crop. So in such a scenario, even though it could be a lucrative options, particularly for those who are growing rice hybrids, to go for a quick ratoon crop, I'm not really sure how far the government will see an eye to eye with regard to growing a ratoon crop. It's still worth considering, at least at experimental level. We should work on it.
Yeah, okay, thank you. I think it's because in US, for example, there are, I mean, it's not in rice, but for example, in fodders or in turf grass. Of course, in this kind of situation, they are talking about regenerative agriculture. So and that is where probably this ratooning or regrowth stuff will come in play. But I agree with your point. I know I have thought a little bit about it, but it's very nice to know more, because you are at that level that you could speak something at the policy level that I would not able to be thinking that way. So thank you very much. Yeah.
Thank you, sir. We have a question from Dr. Abdul Raj. Over to you, Dr. Abdul.
Yeah. Thank you, Sunita. Sir, thank you, sir, for the nice presentation. I just need one clarification. Have you come across any drought tolerant gene having pleiotropic effect on the yield enhancement, sir, apart from having only tolerance?
Yes.
Okay.
Yes, we do have that scenario. We have definitely come across.
Okay. How much yield enhancement you have seen, sir, compared to?
So again, let me tell you, I would not be able to compare that one with varieties like the donors like N22 and Bandhana. That's just simply because of the fact that Bandhana and N22 just do not grow very well in our condition, right? But over cocodri, for example, like the recipient parent, we have seen more than 30% of yield enhancement.
Okay.
Under drought stress particularly. And some of these lines, this is actually just the QTL side I'm talking about, because the genes, the one single gene that I showed, that I have not really translated into the field yet, so I cannot talk about that particular gene. But the QTLs I'm talking about, the one in chromosome 3, when we have introgressed that one, we have seen more than 30% of yield improvement over the recipient.
Thank you.
Are there any more questions? If there are no questions,
Sir, you're muted, sir.
Dr. Sunita, are there any more questions, ma?
no, sir, I checked even in the chat box.
Yeah.
I think
We can wind up today's session. I now request Dr. Padma Kumari to propose a vote of thanks.
So before coming to this formal vote of thanks, I request you to do the honors, sir. So,
Yeah, I just forgot, Dr. Niranjan. When you come out to India, we'll offer you this life certificate. I'll just remove my background. Okay. So it's a proud privilege for us to offer the certificate of honors to you, sir. Thank you very much for being with us. And then despite this late night hours there at Louisiana, thank you very much for being with us and then delivering this nice talk and also enlightening us. Thank you very much, Dr. Niranjan. Thank you.
Thank you, Dr. Sundaram. I'm sorry that you don't need to say me sir or anything, please. I just feel belittled by
Okay,
that.
Dr. Niranjan, thank
Yeah,
you.
that's fine. So yeah, I know I have just come a long way for that. So it's okay. But thank you very much for that gesture.
Thank you.
I appreciate that.
so
I definitely would come to visit if I'm there. There are too many reasons to go to Hyderabad. So
so shall I propose, sir?
Yeah, go ahead, ma'am.
It's my proud privilege to propose this formal vote of thanks on behalf of the seminar committee. First of all, I thank our director, Dr. R.M. Sundaram, for evincing interest to conduct a webinar on this very important burning topic. And I profusely thank Dr. Niranjan Baisak for acceding to our request, and Dr. Satendra played a major role in communicating to him and getting his acceptance. And he has shared his knowledge and expertise. I thank you very much, sir. Having stayed, I think late night it is for you, but still you are with us. We thank you profusely for sharing your knowledge and expertise in this very important topic on breeding rice varieties for alternative irrigating systems through various approaches. The results are very much interesting. I think many of our scientists would take you from this and go ahead with further research in these aspects. Now, I thank the co-host SAR, Society for Advancement of Rice Research, who is a co-host for this webinar. I thank the secretary and members of this society. I'm grateful to all the dignitaries. 72 of them have participated, scientific fraternity, and many dignitaries from ANGRU who have actively participated in this webinar and ensuing fruitful discussions. I thank Dr. Kalyani Barbatika for the introduction, Dr. Sunita for moderating the session, Dr. Girish and Dr. Suvarna Rani for recording the proceedings. I thank Head AKMU and her team for all the resources they have provided for the conduct of this webinar. Thank you, one and all. Thank you. I think now
Can we have a group photo, please? If everybody can just switch on their cameras, we'll have a group photo.
Doctor.
Yeah, thank you everyone. Thank you, ma'am. Thank you.
Thank you, sir. Thank you.
So
Thank you
shall
very
we-
much. Thank you very much, everybody. Appreciate your time, and then
Thank you.
I hope to see you some soon.
Shall we close it, sir?
Yeah.